Over to you, Ken.
All right, thank you, Louise. I will share my screen now. I do ask and do not mind if you have questions while I'm talking, please feel free to interrupt. That way I can sort of address things when they come up, so in case you might forget later. So this is work, well, here's my title: Improving Inference for Nonlinear State Space Models of Animal Population Dynamics Given Biased Sequential Life Stage Data. And this is work that is joint with some colleagues from U.S. Fish and Wildlife Service in California, Leo Polanski and Laura Mitchell. And three general aims of this talk. The first aim is really what the title is saying, which is to talk about how identifiability problems with state-space models are potentially overcome. A couple of issues that we found, at least with our case situation, worked to solve the problem. But this has been an interesting problem. There's been a lot of work, as you'll see. It's a spread over. It's a story that's really spreading over 10 years. I wanted to actually, I'm kind of enjoying this opportunity. I hope I'll enjoy this opportunity to talk about sort of the larger picture that this work fit within. And so, sort of the second point here of science management and laws, to talk about how these state-space models were really a consequence of legal proceedings and have been used. We use them to improve understanding of biological processes and have been used to guide or at least defend some of the managed actions that have been taken. So the motivating situation here is delta smelt, which is a very uncharismatic species, minnow-sized species. It's found only in the California Delta, and I think people can maybe see this hand moving across here. This portion of California is where the Delta is, and there's two big rivers that come and meet there and converge. There's the Sacramento River, which comes from the north down from Mount Shasta there, and then the San Joaquin River, which comes from the south and comes up. And those two rivers come together, meet in the Delta, and then they empty out into the bay and into the Pacific Ocean. They were, say 100 plus years ago, they were the most abundant species in the Delta, but they're now nearly extinct. And that's been the motivation for this work. So part of the reason that the Delta smelt have gone near extinct is because the state of California has been drastically replumbed. And the genesis of this replumbing was the Central Valley Project, which was a project that got established during the Depression when FDR was president. And it was partly a jobs bill in a sense, but it also was meant to generate a lot of power and to provide irrigation water throughout California. And it really didn't, because of World War II, things didn't really get going until towards the end of the war. The first project that was completed was Shasta Dam, which seemed great at the time. It was quite an achievement engineering-wise. It was the second tallest after Hoover Dam at that time. The downside was that it blocked much of the spawning area for winter run Chinook salmon, which has then led to them being an endangered species. The damage that really started being done to Delta smelt, however, was with these giant pumping plants. The first one that went online is called the Central Valley Project Pumping Plant, and it's down near the Delta. And that was a federal project. And then about 13 years later, the State Water Project, which was a state pumping plant, came into being. And these are really huge operations. They are extremely powerful pumps that draw water from the San Joaquin and Sacramento rivers and then deliver it to all parts of California, but particularly to the Central Valley. And these pumps are so powerful that they reverse the flows. In other words, water that's going downstream will start going upstream. That's how strong they are. Not just part of the Central Valley Project, but just in general, sort of the development of California led to a drastic reshaping of this delta area. And these are, on the left here is a sort of an artist's conception of what the delta looked like in the early 1800s. And what you can see here, these are all streams and tiny little rivulets and channels and sloughs and swamps. All this was basically drained and diked until it looked like this now. So it was a radical change in what the environment was. Now, as I said, these waters coming from those rivers, they're the water supply for over 25 million people in the state, and are the central source of water, surface water for Central Valley agriculture. The Central Valley agriculture is big money. The economic value of agriculture in California altogether is about 38 billion pounds, and the vast majority is coming from Central Valley. The draw from water is so great there, the need for water is so great that the surface water irrigation is not near enough. So they've been depleting groundwater for years. And this sign is kind of a famous picture here, where at the top it's showing ground subsidence. So in 1925, the ground was up here. And as of 1977, it had dropped this far. And I'm not sure how far that is, maybe 25, 30 feet, but that's the result of all the pumping. Of course, the other thing that's gone on at the same time with with water use and agriculture was just that the population has grown tremendously. It's near 40 million people now, maybe beyond. And of course, that's meant greater demands on water. So when the Central Valley Project was about to really get going, biologists were concerned, and they said at that time, this could be devastating, and they turned out to be right. But one of the good things that they did was they established these long-term monitoring programs relatively early. And so the first one that went online was in 1958 called a summer townet survey. It was aimed at monitoring striped bass populations because that was of more interest to them. And that was right after this Central Valley Project pumping plant went online. And then the next big survey was the Fall Midwater Trawl Survey that started in 1967, again aimed at striped bass, and that was when the State Water Project came on board. And then 3 more surveys were added, and the thing to note, in 1992 and then in 2003, surveys were established in particular to monitor delta smelt because they were starting to see severe declines in delta smelt populations. And so in a way, it's a pretty amazing data set, and that we've got right here. We have 5 different surveys that have gone on, some of them for over 60 years now. Well, one of them over 60 years. So it's quite a data set. This is a picture. These are plots of, on this left plot, this is showing the water exports on an annual level from the combined pumping plants, Central Valley Project and State Water Project over time. And is I think 1955 or 56 when the Central Valley Project went online, and then in 68 or so the State Water online. That's why you see that big jump. And then you can see what the draws have been like over the years. And sometimes these big declines here are more due to drought than anything else. This right-hand plot is the same thing with the exports, but the red lines are showing indices of the delta smelt abundance. And right about 67, well, that 68, that's the State Water Project going online. And that was just before that really went online, that was one of the largest indices values for delta smelt. And then there was another, then they missed a year or two, and then there was a big spike again. But then since then it dropped down quite a bit. And it was this period right here that was very, very worrisome to people. They thought, well, this population's in trouble now. There was a bit of, and then some actions were taken. And a bit of resurgence here, probably more due to good rain years than anything else. And then a drastic decline with a spike in 2013, which was another really good water year.
Ken, are those there's enormous variability there, especially like even in the 70s. Is that like real variability in delta smelt numbers? That very early period where there's the first 3 or 4 years, and then it bounces up to more than double in 1 year. Can fish do that?
Yes, they can. That's probably there. Obviously, there is sampling noise attached to this, but actually, this is probably more environmental variation than anything else. They're
Right.
predominantly an annual species, and they're very much a boom or bust species. And if you have a good water year, you get good reproduction, good survival. You can see a spike like this. And we saw that on a smaller scale in 2013, where here the population's really in serious decline, you can see. And we had one great water year, and they came back and then returned to normal. And now they're very bad shape. So yeah, these are reflective, probably more of the biology and good environmental conditions, and not that much sampling variability.
Thanks.
These are pictures across time. The x-axis is showing years. These are indices of delta smelt from multiple surveys. Some of the ones I mentioned, these top 1, 2, 3, 4, and then here's one down here. I mentioned those previously. They're all showing the same picture as you go through the years, particularly from 1980 on. You can see relatively steady declines in the abundance of delta smelt. And these different surveys are capturing fish at different life stages. Same story. They show a population in severe decline. So the severe declines that they saw in the late 80s led to endangered species listings at the federal and the state levels. And along with those listings then came management actions to attempt to save the population and help it recover. And these management actions primarily focused on two things. One was to slow down the amount of water being pulled out by the pumps during January to March, because that's when reproduction, spawning tends to occur. And where the spawning happens to occur is relatively near the pumps as well, the major pumps. And then the other management action was to increase outflow to the delta during the spring and during the fall. And what this meant was to release water from upstream dams like Shasta Dam and other upstream dams to increase that outflow. And the fish, they stay in the delta, but that fresh water coming through appears to increase their survival. And that's what our modeling showed as well. So with this endangered species listing, these management actions that cut back on the amount of water being exported, there has been a lot of political controversy. Some of you have probably heard about this. It's been going on for decades now. And Sarah Palin, who was the vice presidential candidate for the Republicans back in 2008, I believe, she came to Central Valley later, and she was from Alaska, where they have very large fish, salmon and so on, and halibut. And she got a lot of claps from farmers about statements like, why are you protecting a 3-inch fish? Where I come from in Alaska, we call that bait. There's no need to destroy people's lives over bait. So that got a lot of airplay. And then just 5 days ago, Trump again was talking about this. He's weighed in on this a few times. And again, he's saying California has to ration water because it dumps so much in the sea to help certain little tiny fish, and that it's ridiculous that they're taking this water and shoving it out to the sea. So it's still very politically hot topic. So one of the consequences of these management actions was, there were several court cases, but the really big one was in 2010, where the water users, there's multiple irrigation agencies, sued the federal government over these restrictions. And this was quite a circus in some ways, you could say. It was a kind of amazing, intense 1-week period of testimony in court hearings where the plaintiffs, the water users who have a lot of money, brought in several, very well-known, very reputable statisticians and fishery scientists. Rick Doriso, Ray Hilborn, Brian Manley, and Ken Burnham were brought in as expert witnesses. And then on the defense side, they had a biologist from I think Fish and Game at that time, and then Bureau of Land Management, and then they brought me in as well. And the judge had his own expert witness even. It was Andre Punt, who some of you may know. So it was pretty interesting to see this sort of confluence of people. And the testimony from the case, you can go online and find the judge's decisions here and comments here. That the gist of these comments here, coming from Rick DeRiso, Ray Hilborn, and myself, was that we all agreed that really we needed a life cycle model for Delta Smelt to evaluate the effect of management actions and out from environmental factors. And during the proceedings, there were some statements by some individuals that a life cycle model could be built, say, over a weekend. And it certainly, I certainly didn't do it that fast. And
a long weekend.
Yeah. So, but one thing, and I didn't realize this until I read the testimony again late, just a couple of days ago, that Hilborn had worked with Mark Maunder. They'd gotten together to try to build basically a life cycle model, and then they quit. They abandoned that effort as too complicated and too time consuming. So I thought that was an interesting remark. So that there was that sort of 1 week of intense testimony and court hearing, and the judge, Oliver Wanger, then came to his decision, and he said that, and these are really strong words with regard to management actions. He said that the way that the U.S. Fish and Wildlife Service had administered this protection plan under the Endangered Species Act through this biological opinion, that just means management actions, basically, arbitrary, capricious, and unlawful. And those are really bad words. And I wrote these words on a sheet of paper and I put it above my computer when I was working for Fish and Wildlife Service, just to remind myself, am I being arbitrary, capricious, or unlawful? So, and then he said, the public cannot afford sloppy science and unidirectional prescriptions that ignore California's water needs. And this was in the New York Times, so it made a lot of coverage. Now, the sequel to, well, let me finish there. This is more from Judge Wanger. He said, he concluded that a quantitative life cycle model is the preferred scientific methodology. Fish and Wildlife Service made a conscious choice not to use the expertise within the agency to develop one. I was working for them then, and they actually talked to me about doing this, but I was busy working on salmon, and so I didn't get involved. And that's why they were actually able to, they brought me in as an expert witness, because I actually had not been involved with the development. But the judge said, we can't really force the agency to create one, but Fish and Wildlife Service said, given all this, let's go ahead and let's do one. Let's make a life cycle model. Now, the follow-up to the story, which I don't have a slide for, was actually Judge Wanger's decision was overturned in the Court of Appeals, but they said that he had sort of not done it correctly, you could say, but we decided it was important, and let's go ahead and do it.
Did they say the judge was being capricious, making his original decision?
No, I don't think those words were used. So I really have a lot of respect for Judge Wanger. I was very impressed myself how quickly he grasped what was going on. And I've recently found a video from him that 8 years later, 2018, YouTube video of him talking about this case. It's like one of the biggest cases in his sort of legal history, I think, and he still feels, I think, kind of sore about the appeals decision. But and he said, I think I know as much about Delta smelt as a lot of biologists do after working on this for 20 years. And I tend to agree. I think he actually was a very fair judge in a lot of ways. So here was our plan after this court decision. Let's put together a team of individuals. So we brought together biologists, ecologists, hydrologists, and statisticians. Let's sit down and talk about all the factors that probably are responsible for the demise of these fish. And I've listed some of them here. Then let's formulate a model, and then let's use the data that we have as fit models, evaluate them, compare them, and then use the model. So we thought, well, we'll have this done in a couple of years maybe, and we didn't get it done that fast. So first, just talking about the data a little bit, we have a lot of data, as I mentioned earlier. And the nice thing is that these data are taking samples from the fish at different life stages. So if we start in February, March, that's when spawning takes place and the adults are out there. And these fish primarily live for 1 year. Then we have a survey that catches these adult fish. There's two surveys there. And then as you go through time throughout the year, these fish are hatched, they hatch, and then they're larvae, and then they grow into small fish. And we have another survey here, the 20 millimeter survey, aimed at delta smelt that catches some of them. And then they grow some more and become juveniles. And we have another survey called the summer townet there. And then they survive through the summer and go into the fall, and then they become what are called subadults. We have a fourth survey here that's catching them. So we have year-round sampling. In this picture here, which is not the easiest to see, this is showing the estuary, the San Francisco off to the left. This is the delta. All these little numbered points here are sampling locations for one of the surveys. So some of the surveys have like over 100 locations being sampled once a month. Others have like 50 being sampled every 2 weeks. So it's pretty good temporal and spatial coverage. And this is just a picture of one of the nets. That's the 20 millimeter gear that's used. Now, so we have this sort of abundance of data, but the problem was that they all have different efficiencies. They have different probabilities of capturing fish. And really nothing was known about that. And we thought that we really needed to make some sort of adjustments to account for differences in efficiency. So we proposed this study and then had this study implemented where this is part of the Sacramento River, and these are like lanes that boats would go down pulling different types of gear, and then we would randomize the order that they would go in, and we try to keep the tow lengths the same and so on. So they were in a sense meant to be fishing the same density of fish. So this provided a lot of gear information that had not been collected before. And from that, then we used that data to fit gear selectivity curves, and we used a pretty well-established technique by Russell Miller called SELECT, which the underlying statistics on this is that the catches are multinomial. And we made slight modifications in their formulation, but to fit basically multiple gear efficiency measures for the different gear types. And so the probability of capture was a function of fish length, and this P gear had to do more with like the volume of water that was being sampled than anything else. So this is more like how efficient is the gear and how much water is being sampled. Combine those things, put those into a multinomial, use maximum likelihood to estimate the underlying parameters of the gear efficiency relationship. And these plots here are showing estimates of gear efficiency for four different surveys. All right, so you can see here that this is called the smelt larval survey, the 20 millimeter. These tend to catch smaller fish, and they have a much higher probability of catching small fish than they do big fish. Big fish are thought to be able to evade the net or simply sort of bounce out of the net. They're just too big. They're not going to get stuck in the mesh. And then the larger fish are caught with higher probability with the summer townet. And then this gear here is considered almost 100% efficient for larger fish, the way it works. So this was really useful information and has helped us then for the next stage of the work, which was to then use those gear efficiency measurements and the historical surveys to come up with abundance estimates, or at least abundance indices. And the underlying estimation strategy was basically stratified random sampling, where we used ratio of means estimators in a design-based setting, but it was model assisted in the sense that we had these gear efficiency modeling results. And the gist of it was that for a given stratum, a given life stage, year, month, year, and stratum H, what we're doing is we have measurements of the volume of water that's in a stratum within a different particular region, and we multiply that by an estimate of the density of fish. So in principle, it's a pretty simple idea. It's just volume of your stratum times the density, an estimate of the density of fish in that stratum. So this is what we were building, collecting these data for, was to put together a cycle model and do it within a state space model framework. And I'll say a bit about this, but for many of you, you know about state space model, I'm sure. And the gist of it is we have two time series running in parallel, where the state here is the unobserved variable. This could be, say, delta smelt abundance over time. And then the observations would be like our catches from our surveys. All right, so the gist of this is that, say, abundance at month 1 is a function of abundance in the previous month, and so on. It's a first order Markov process. And then the observed samples are functions. So y, like catches, they're observed functions of the underlying hidden state, which is the abundance or the density of those fish. Now, state space models are used for lots of different species nowadays for population dynamics and for movement and so on. And I mostly put this picture in here because I have charismatic species envy. I'm working with this little fish up here, but there's lots more charismatic species that people have used these for, polar bears, and I'm probably mispronouncing this as Ferragunus owls. I'm sure Richard could correct me on that. And then this is a type of mongoose. So all 4 of these species have been modeled with state space models. And so state space models have been around in ecology for quite a while. It was really in fisheries work. They first got used a lot in the mid-1980s. And a quote from a paper by Marie Auger-Methe is that they've become the favored statistical framework for modeling animal movement and population dynamics. And a main reason for state space models' popularity is that they separate out process variation. So by this, I mean environmental, say, year-to-year variation in dynamics from the observation error, the sampling error. So the most basic state space model is called a normal dynamic linear model. And here we have that first order Markov evolution of the underlying states. And the distributions are assumed normal, and the relationship with past values is linear. And then likewise, the relationship between observations and the hidden states is also normal and linear as well. And the big distinction here then is between the sigma squared p for process variation from sigma squared o for observation. And so again, that's one of the primary attractions to ecologists, is to be able to separate out that natural variation from the sampling variation. Now, that looks good. It seems like a great idea, but there are problems with state space models, particularly these normal dynamic linear models. And I want to back up and say part of the reason for these normal dynamic linear models being very popular is that it's relatively easy to estimate the parameters, and the algorithm that's used to construct the likelihood is something called the Kalman filter or the Kalman smooth. And those are just procedures basically for coming up with the marginal distribution for the observations, and then you can maximize that. So relatively easy to do that. So they're relatively easy to fit, but there's serious difficulties and problems with them in practice. And Brian Dennis and others has emphasized this particular problem. And the underlying problem is it's difficult to separate out and distinguish this process variation from this observation variation. And this is a picture from a 2006 ecology paper showing the profile likelihoods for, I can't remember if this is simulated data or real data, where this is the profile likelihood, this is the observation variance, and this is the process variance here. And you can see how flat these profile likelihoods are with regard to these two parameters. So you don't have much information about what the value is. I mean, 0.04 is just as reasonable really as say 0.01 for the observation variance in this. And then we have a long flat spot for the process variance as well. And then if you look at the joint profile likelihood of the two, they're highly correlated with each other. And so, for example, we're trying to say minimize the negative log likelihood. Here we have 2 points. There's another one over here, I think it's kind of hidden away, where both these values, both these combinations are yielding relatively similar negative log likelihood. So what we have like a bimodal distribution here, and either combination will work. We could have, what it turns out to be is you can have a trade-off where you can have, I'll say, a high observation variance and low process variance. And the other way around, you could have a low observation variance and then a high process variance. And you really can't distinguish them, which is better in terms of likelihoods. Criticism of state space models. And also because of this, if it turns out there's been a lot of other work on this, that if you're trying to detect, you have models that include density dependence, you often wrongly say there's density dependence, and it has to do with this identifiability issue. And this has been explored. And so you're more likely to say there's density dependence when in fact there is not. Another issue, well, this is sort of a systematic problem brought out by Maria Agarmethi and others, is that these process variance estimates in general tend to be biased high, whereas the observation variances are low. And so this is important to remember. These are results that they have observed with normal dynamic linear models. We're going to see that we actually get different results later using non-normal and non-linear state space models. These graphs, these box plots down here are showing, for example, I think, yeah, it's over here. This is the, these are the process variances or standard deviations, and these are box plots of estimates. And the red dot is the true process variance. And these box plots are showing how that bias, there's bias in those estimates as the ratio between the observation noise and the process variation changes as it increases. So this bias, though, in this case is pretty consistent, no matter what the combination is. So how do we deal with this problem? The underlying problem is say weak identifiability, or sometimes called parameter redundancy. And there's 3 different general approaches to dealing with this. One is sometimes you can reformulate your state space model so that the process variation and the mean in that state transition equation, the first order Markov equation, depend upon the same parameters. So you're getting rid of a parameter and you're sharing it between the mean and the variance. And that's one way of handling the problem. Another way to handle the problem, which I like very much, recommended by Brian Dennis and others, is, well, if you can take duplicate observations, if you can get replicates, then that's ideal. But that's not always feasible. A third approach is that you simply are able to construct independent estimates of observation error. And this is something by Jonas Knopp and others that have promoted. And this is what we end up doing with ours. We have independent estimates. So our questions were, how much, so we know these problems exist for normal dynamic linear models. How much of a problem are they if you're dealing with a non-linear, non-Gaussian state space model? And in fact, what if these state processes are functions of covariance? So you've got the state processes are being modeled by functions of, let's say, water flow or water temperature or salinity. We still have these identifiability problems showing up here when we have this kind of sort of more complicated state space model? And then the third question was, do these independent estimates of observation error help with these more complex state space models? Knopp said, yes, they help with the NDLMs, but do they help with these more complex models? So this was the model that we ended up with here. And it might look somewhat involved, but it actually is not that complicated. What it does, it parallels the life history of delta smelt. And this is the state process model for each of 4 different life stages. Post-larvae, those are the ones these are like the young that have just hatched. The juveniles, these are the young that have grown up a bit. And then the juveniles grow up more, and then they become adults. So you have 4 life stages, and we link them all to the previous life stage in terms of parameters that reflect either reproductive success. So this row of T is talking about basically number of young produced per adult. And then these next 3 parameters, these phi's, are all survival probabilities. This is the probability of living from post-larval stage to juvenile stage, from juvenile stage to subadult stage, from subadult stage to adult stage. All right, and we modeled these processes as a random variable. So sort of this is a hierarchical model here. So for example, we're saying that recruitment, we're modeling recruitment, log normal recruitment, or we're modeling recruitment on the log normal scale as a function of covariates, where the subscript R refers to recruitment. So we have a vector of covariates. And then it has its own process variation for recruitment. And then we just repeat that for the other 4 processes, just changing from, here we're talking about some value that's going to be, well, greater than 100 typically, like the number of fish per adult. And these are then values. These are survival probabilities. They have to stay between 0 and 1. So each of these processes has their own process variation. And the mean structure involves potentially different covariates. The covariates are measured at different points in time. So we may have the same covariate showing up for multiple processes, let's say water temperature, but we're talking about the water temperature during the recruitment period, or versus the water temperature versus during the, say, months of April or May. Then the observation model is based upon those survey data from 4 different surveys, and we link them to the underlying states with a log normal distribution. And these variances here, these are our estimated variances from our abundance estimate work, from the abundance estimates work. So these are being estimated independent from the model. So a bit of a 2-stage procedure. We estimate these outside the model fitting, and then we just plug them in. And here I'd say that these observation variances are all externally estimated. And one other thing to point out here, we know that we had problems with the gear efficiency estimates for 2 of these surveys, the juvenile measure and then the subadult measure. This is the summer tonet, and this is the fall midwater trawl. And we were convinced that we had biases present that we just did not figure out how to correct for with our estimation. And that's why we called our abundance estimates for those time periods indices rather than estimates. So here we're saying these are estimates for the post-larvae and adults, but here we're saying these are indices. And that the index part is being reflected by these 2 psi parameters that are bias parameters. Okay, so want to say a bit about formally looking at parameter redundancy in state space models. And this was something that came up later in the work, and when we learned about this. The gist of this work is based upon work that was done by Catchpole and Morgan with a series of papers in the late 1990s, which present a very nice way of determining. They call it parameter redundancy, or you could just say unidentifiable parameters. There's some distinctions that are important. But what helped us most was this 2016 paper from Diana Cole and Rachel McRae, where they focused on applying these techniques from Catchpole and Morgan to the problem of state space models. And without going into details too much, well, first I want to say this was a very useful paper for us. It included Maple code because they use symbolic algebra to do the determination of what parameters can be estimated and which ones cannot. But here's a very hand-wavy way of saying this, and I know it would make some of these folks cringe to say this, but the experts in this area, but to some degree, this is a little bit like a method of moments notion that if you specify the expected values or the expected value squared of observations, and you write them as functions of the parameters. Then equate these functions to observable sample moments, and then see, can I then have a system of equations? Can I solve for these parameters? If I can, then that's one way of showing that I have identifiable parameters, if it's not redundant. Their technique is far more sophisticated than that, but I find it handy at least to keep this in mind. And so this table here, I just want to show that we used the Colin-McRae method in the Maple code and applied it to our sort of what was called our full model, which had 18 parameters in it. Well, no, this is a much smaller version. I guess it had a total of 8 parameters. And we looked at whether or not covariates were included in modeling the state process. We looked, here's the parameters we wanted to estimate. So here's 9 different models. Here's the parameters in each of the model. And this was the result of the Maple code telling us how many parameter combinations could we estimate. And once we got up to this 9th model here, we could estimate all 8 parameters. But for these others, you can see here, like here's 8 total parameters, we can only estimate 7. Okay, and these are combinations sometimes. And in a lot of cases, you can only estimate singleton parameters. So here we can estimate, let's say, eta naught and eta 1 separately. But in other cases, maybe all you can estimate is eta naught times eta 1. So that would be one of the combinations that you could estimate. Okay, so we wanted to see just how well our model could sort of reproduce the results if we're generating directly from the model. So we did an extensive simulation study. And we looked in particular, and we did this all in a Bayesian framework. We were using JAGs, and we looked at 2 particular situations. One is that we plugged in these fixed observation variances, and the other is that we then estimated those observation errors internally. Okay, so we were not plugging in these external estimates. We wanted to see what the difference would be. And then we looked at posterior distributions for parameters and states. And one of the things that we learned here, which was very useful for us, was that whether or not one plugged in fixed externally estimated values for observation noise, or they were estimated internally, they really had very little effect on the estimates of the process coefficients. And I'll just focus on one box plot here. These are box plots showing, well, I guess it's what, 24 different parameters. And the blue dot here is the true value. And then the box plot here is showing what the, I'm sorry, there's actually 12 parameters. This row has the observation error fixed externally, and this has the observation error estimated internally. So that's the distinction between this top row and this bottom row. So if we focus here on this phi pi L, so this is the larvae survival, and these are 3 parameters involved in that model for survival, and the blue dots are the true values, and these box plots are simulations estimates. And we can see that, well, first of all, if you contrast this top row to the bottom row, there's very little difference. So this is externally plugged in, this is internally estimated, very little difference in the quality of the estimates of the process coefficient. So that was very reassuring. And so it looked like we could estimate process coefficients fairly well in either case. So that was one thing that was encouraging. The second thing is we looked at then what's the effect on the estimates of the process variances, whether we plugged in externally estimated values of observation noise or we estimated internally. They both had the same problem in the sense that we ended up with estimates of process variance that were biased low. Okay, for all 4 processes, this is recruitment, survival, survival, survival. And in every case, we tended to underestimate the true values. The blue dots are the true values, and these are box plots of the distribution of estimated values. And you can, this is the median line. You can see we kind of, with some, there's one exception here, but in general, we tended to underestimate that. And that's in contradiction, or that's different than what Maria Agar-Methé found with their NDLM work. The other thing that we looked at was, well, if we estimated the observation variances internally, how do they compare to sort of the true values? And when we did that, we found that they were biased high. So this is kind of the flip side of what was seen with normal dynamic linear models. We're tending to underestimate process variance. We're tending to overestimate observation variance. So that was interesting to observe. Another set of parameters we're very interested in was this bias in the observation models for juveniles and for subadults. And this was another reassuring result. We found that whether we plugged in these externally estimated values for observation variance or we estimated internally, in both cases, we got accurate estimates of what that bias was. One distinction that we did see, though, was that if we estimated observation noise internally, we had less precision in these estimates of the bias. But that makes sense in that you are spreading your data more thin, you could say. You're trying to estimate more parameters. So expect when you estimate more parameters, you're going to have less precision for them. So that was a sensible result. So here was our application. We went back 20 years with this work to fit the model. I think it's running from 1993 or 4 to 2012. And the reason we picked this date, these were the dates that we had, most of the surveys were in place that provided information about these 4 different life stages. Future work, not by me, would be to go back further in time because we have such a long data set, but then we have fewer surveys. So we tried to make our work a little bit easier by just working with, you could say, more or less complete data sets. So we fit these models for these different processes, and we had quite a few different covariates that were tried for each of the processes. And these things reflect environmental conditions like water flow, that can be manipulated by dam releases. Where the salinity, the 2% salinity line was, it turns out it's a manipulable thing in the delta, and it has a big effect. If the salinities stay above 2%, it turns out that's very good for delta smelt survival. And so they try to manipulate that line and push it out towards San Francisco. So these covariates here represent management actions. And then there's some things that we have very little control over. We don't have that much control over what the prey are, like striped bass or prey that eat delta smelt. And then we have some fish that are competitors. They share sort of the same ecological niche. And then here was another factor that is sort of human control, and that's the amount of water that's flowing out as a function of the pump management or pump actions. So here's the results with these real data. And this is a contrast here between, we're talking about process variance. It's a contrast between internally estimating the observation noise versus plugging in those external estimates. And what we found is that when we do not plug in the observation variances and we estimate it internally, we see tremendous declines in process variance. So this corresponds to what we saw with the simulation results as well. And these are quite drastic reductions in some cases. So the process variance is going down, is decreasing 81% compared to what it would look like if we plugged in external estimates of observation noise. And so this has important management implications because if we estimate internally, this is indicating that we don't have as much environmental variation as perhaps maybe we do. So there's still a question of what's right or wrong here, but the main point is that there's quite a difference here as to whether or not you plug in these external estimates or estimate them internally. So it has important management implications in terms of, say, how sure you are about the effect of your management actions. And again, as for the simulation study, we saw the process coefficients are not too badly affected. So these are coefficients that reflect the effect size of different covariates. And then lastly, oh no, there's still one thing on bias, sorry. When you look at estimating observation variance internally, those estimates are larger than those fixed values. And again, that matches with what the simulation study showed us as well. Yeah, here's the last parameter that we looked at. We looked at these bias parameters for those two different surveys. Psi j is for the juveniles. Here it means summer tonet, that's juveniles. And then the bias for the subadults, that's the fall midwater trawl survey. And the effect of using internally estimated observation variance really didn't have that much effect on the average value or the point estimates of these bias parameters. The point estimates, say for summer tonet, it's about a 50% bias, about half the number that are out there. The estimates of abundance are about half of what's truly out there. About the same whether or not you use external estimates of observation variance or internally estimated. But the difference that's most noticeable here is that there's more uncertainty, there's more variation. And again, it's that principle of where you're trying to estimate more parameters, so you expect your precision to go down. Yes, and then another, and this is what is of most interest to managers and biologists, was this notion of if we manipulate or can manipulate certain environmental conditions, what's the effect going to look like on different processes like survival? So what these plots here are showing are estimates of recruitment on the y-axis versus varying water temperature. Okay, so for example, upstream releases from dams, like from Shasta Dam, they tend to release cold water, and you're able to then to lower the river temperature. And the question was, so if we could get lower temperatures from say 14 degrees Celsius to say all the way down to 17, how much of a gain? Oh no, sorry, this is an increase. I got it backwards, sorry. This is an increasing temperature. So this is going from cold to hot. So as the temperature increases, this is showing what how the recruitment rates may drop off here. Okay, so up here we're about say 38 post-larvae per adult being produced at these cooler temperatures, and we're dropping down to, I don't know what that is, 12 or something like that, if the water's that warm. So both cases where we use externally estimated values of observation noise and then compared to internal, the overall pattern's the same. That's because the covariates, the coefficients are not affected so much. So we get the same general pattern, but the precision, or you could say the prediction error, is higher when we plug in these, when we're using these external estimates. And again, that could be due to the fact that it looks like these estimates of observation noise maybe are too low. Or no, yeah, okay, that's what the effect is. They tend to be too low, but anyways, you're getting apparently more precision. So that means better prediction results, perhaps artificially, when you're estimating that observation noise internally. And then this is sort of a final big picture look at the results of the model that in a very sort of almost cartoon description. So these are summaries of sort of the dominant covariates for different life stages. And so this top one is recruitment, and in a way, we say a lot of obvious things at times here, I must admit. It's like water is good for fish, hot water is bad for fish. And that's really what the end result is in some places, but at least we quantify what the relationship is. So this is recruitment here. And as I showed in the previous page, this is saying the hot water is bad for recruitment. Okay, so the survival of post-larvae to the juvenile stage increases as the inflow into the delta increases. So that's what this plot is saying right here. This is showing how juvenile survival, or sorry, post-larvae survival increases with increasing flow into the delta. And so we can quantify then, if they could bump flows up to say, I forget what these measures are now, if it's 750,000 or 7.5 million actually cubic I'm not sure what the measurement was, meters over a month or something like that. I can't remember now, but it's showing actually quite a change here, quite an increase from almost like 0.75 survival compared to 0.25 when it's down here. So to the degree that you could control outflow, you definitely want to push it up if you want to increase survival during that life stage. Sometimes you can do that by manipulating the AMs, but other times because of drought, there's nothing you do, but at least you know what's happening. You have some idea. And then here's looking at survival at the next life stage. And we really found very little relationship here. We couldn't find covariates that mattered much. We found that if the water was slightly saltier in the delta, that we were actually getting lower juvenile survival, but it's almost non-significant here. And this is actually at odds with what the biologists believed as well. This is contradicted biological reasoning. And then lastly, this is an important life stage. This is the probability of fish surviving from subadults to the adult stage. So this controls how many fish are around to reproduce. And this we found to be a function of quite a few manipulable things. This is called Old and Middle River flow. This is the amount of water that basically was being diverted to pumps. The Secchi has to do with the turbidity of the water, how delta smelt tend to like more turbid water, or they survive better, what I should say. Well, they tend to go for it. They tend to move there. They also found through lab experiments with aquariums that predators have a harder time finding them in the muddy water. So that makes sense. And then here was an interaction between the two. And the gist of this was very common sense too. If we could sort of have that water going away from the pumps, that survival rate increased as well. So it was reassuring to see that our statistical results matched biological theory most of the time. Another thing to show here is that this is sort of the comparison between doing state space models and not doing state space models. We could calculate recruitments, juvenile, post-larval survival, juvenile survival, and so on, sort of naively, just by looking at ratios of abundances between successive life stages. But you can get ridiculous estimates. So, for example, this is the ratio of of juveniles to post-larvae. And that ratio, this is for 1995, this is a naive survival estimate of almost 250%, which of course doesn't make any sense. But that's what happens in a non-state space model framework if you're not kind of nesting and constraining those estimates. Whereas the state space model always will yield a reasonable estimate. The survival values will always be between 0 and 1. So that was another sort of a plug for doing the state space model work. So just a conclusion with regard to what we've learned here, that from both the simulation study and the case study, if we internally estimate the observation noise, it doesn't have much effect on the coefficients for the covariates of these processes. Good news. The other thing we learned from simulation is that when we plug in these estimates of observation error, and I didn't really talk about estimation of the states, the abundances, but we got more precision there as well, and some of the parameters are estimated more precisely as well. The thing that simulation also revealed, that we're able to accurately estimate observation bias no matter what. And that was really nice to know, because if you're working with normal dynamic linear models, it's extremely difficult to be able to separate out, say, bias in the observation model from, say, a covariate or term that's actually in the process model. So you have sort of identifiability problems that will show up between a bias parameter in the observation model and some multiplier that's in the process model. So that was very nice to see. And then lastly, we saw from the case study that plugging in these if we estimated observation errors internally, the process variation would get smaller. And again, that's opposite to what has been seen with normal dynamic linear models. Okay, almost done here. So the last thing, there's been a lot of continued work that's been spin-off work. There's a subsequent work that was done led by one of my colleagues, Will Smith, where he and I was a co-author with Laura Mitchell as well, where we looked at can we use state-space models to better understand the effect of the pumps on mortality of fish within the delta. And it wasn't a life cycle model so much, but it was a hierarchical model. And we were able to tease out to some degree with some strong assumptions, sort of the direct mortality. That's fish actually getting drawn into the pumps and of course dying there, versus indirect mortality, which means that there's kind of trapped from the hydrodynamics to stay in this area of the delta that has a lot of predators. So we could roughly break that apart. So that was kind of nice work. Current work that Leo Polanski is leading on right now is to do population viability analysis with this model and to do a lot of what if scenarios. So what if we could control outflows over several years? What might we do to the overall population growth rate? A third spinoff from this, which was really rewarding to all of us, I think, was that from our gear evaluation studies, we found out which gears were better at which life stages. And so we said, well, we ought to be using the 20 millimeter gear like much more of the year, not just for the just emerged salmon, or smelt, but also for these larger sizes. The gear actually does pretty well for fish that were larger than they thought. And then at the other extreme, this Kodiak trawl gear is very good for not only the large fish, but slightly small fish. So we switched to a program. This is a brand new monitoring program that's been in place for like 4 years now, which goes year round, and we just switch the gear that's used for the different life stages, but we're only using two types of gear now. And that's been very rewarding. It's we randomly picked, we used generalized random tessellation sampling, the GRITS design to locate the sampling location. So we kept moving them around. That's something I didn't say about our monitoring design, the historical monitoring design. Those sites were fixed. They were picked once, like in 1958, and they've not moved since. So that's obviously a weakness of those designs. Whereas this new monitoring program is randomly varying the locations all the time. And the population's in trouble, and I don't know, it may go extinct. And the fall midwater trawl, which has been sort of like the traditional main survey for Delta smelt, caught no fish at all in 2018 and 2019. But reassuringly, this enhanced Delta smelt monitoring program this year has caught 30 fish, which tells us they're not gone. There's not many left, but they're not completely gone. And with that, I will conclude. And this is just, these are some of the papers that have resulted from this work. So thank you for your time, and open for questions and comments.
Yeah, thank you very much, Ken. That was really interesting, and especially to hear about the whole story behind this work. Does anyone have any questions for Ken?
I've got a non-scientific question. So there was that court case in which you were an advisor, and then you built this model. Was there any follow-up legal action on this that used your model?
Well, that's used the state-space model, you mean?
Yeah.
Well, I guess in a way the good news is we haven't been sued for that yet. They haven't been sued yet. So you could say, well, see, it took so long to get done that the judge is long since retired, and we're just kind of waiting for people to disappear and not forget what we did. But the short answer is, actually, I think it's good that I think we've put together something that's defensible enough. It's kept them out of court anyway. They haven't been sued for arbitrary and capricious management actions since then.
That's got to be a good thing.
I think so. The bad thing is that the population is still appears to be going extinct. So, and I would like to talk to Ray Helborn again sometime about this, because I remember it was either him or Rick DeRusso saying in court, well, this was something that he felt like with a graduate student and a good weekend could probably put something together. And I know that I'm slow, and it took me about 8 years, but it'd be good to have a conversation about that now.
So, sorry, David.
No, go ahead.
As you might expect, I've got a few, but I'll try and restrain myself. I thought it was interesting, just a comment to start with. I thought it was interesting that the process error goes down when you estimate the observation error in this case, sort of implying that for a naive person looking at this, like a manager or whatever, if you throw some information away, if you pretend you don't know the observation variance, you get better, as in more precise predictions. So it must be very frustrating to them for you to say, no, you need to put the known observation errors in, or the estimated observation errors, and then suddenly your predictions are much less precise. But the actual question is, do you have any sort of intuition about why in the normal linear model we're getting one result with bias in observation versus process error, and in this case you're getting the exact opposite when you try and estimate both?
Short answer is no. I mean, I've thought about it, and I've wondered how to even try to arrive at an explanation. And I haven't come up with one yet, so I don't know. And that's, I mean, this is, you could say, well, maybe these results are very specific to this model as well. So if you do another nonlinear, so it's, I think the jury is out, so to speak, on this. There's definitely more work to do here, but unfortunately, I don't have an answer on that.
Okay, just to check one thing off the list. I noticed this was Bayesian, and I guess Marie's stuff is probably classical. So, and you didn't say anything about priors on the observation error parameter or on the error parameters. Right.
Well, first of all, Marie's work was both Bayesian and frequentist in that paper, that 2016 paper. She did both and came up, and she used JAGs and came to very similar conclusions. The priors were for the variances, they were gamma type. They should be technically, I guess, well, it doesn't matter that much. I say you have inverse gamma. I think we use gammas for the priors for the coefficients in the process model, the beta knots, the eta knots, etc. Those, we used that technique, which is kind of like a logistic distribution, with that idea that the induced prior on survival is relatively uniform and not bathtub shaped.
So
I think, go ahead.
The difference in result, I guess I was interested in the observation variance estimates, or the process and observation variance estimates, and those aren't influenced by the priors effectively. I mean, it's uninformative in some sense.
Yeah, actually, I think we also tried just plain uniforms, which is kind of like something close to what Gelman has suggested as well. And we didn't, it's been a while, I don't remember anything sort of being really apparent about that. So, but we did do, I think, due diligence, let's say, on sensitivity analysis, and didn't see much happening there. We had a lot of data. I think that's one thing. This was definitely a case where data swamped the priors, I think, in most cases.
We had a hand up from Michael. Do you want to go ahead, Michael, to ask?
Yes, thank you very much for the nice talk. Actually, my comment is a bit similar to Len's comment. So first, I wanted to say that I'm a big fan of the Catchpole and Morgan method for identifiability, because it also allows you to find out the estimable combinations of parameters rather than just parameters. So, and of course, the extensions by Cole and McRae. But I was a bit, I was thinking about the Bayesian implementation of this, because of course, the effect of the prior on identifiability can be quite important, both because, of course, of the direct information that sometimes we can bring in through the prior, but also because sometimes our prior distributions, they induce dependence between the parameters, between the model parameters. And of course, this prior dependence between this dependence between the model parameters, it can affect identifiability also. So in this sense, I was wondering if what the Bayesian model was doing was slightly different than what the Catchpole and Morgan method would indicate.
I don't know. I'm glad you raised that point. I hadn't really thought about things that way. I mean, we did look, but you're right. I mean, we used independent priors for everything, and pretty much across the board. But then the posteriors are dependent, and obviously priors can compensate for or sort of overcome to some degree identifiability issues. But we tried to pick such sort of weak priors that we didn't think we were doing that very much. So I don't know if I've really answered your question. You want to repeat it again or make sure I've answered it?
No, yeah. So for example, yeah, if we're looking at the generalized linear model, the quite common G prior induces quite a lot of dependence between the model parameters, and it can affect the identifiability of the model parameters, although the G prior is supposed to be a non-informative unit information kind of prior. So yeah, this was my point because the Bayesian setup may change a little bit what Catchpole and Morgan outcomes are.
Yeah, it'd be a good thing to explore more, and we didn't explore that very much. So, yeah.
Yeah. Thank you.
Mm-hmm.
Okay. Are there any more questions?
Can I have another really quick one?
Okay.
Promise it's quick.
Okay.
What about model selection for all these covariates? I mean, how did you do that?
Yeah, we did. We did do that. The general approach that we used, and it's slipping my mind right now. We didn't use reversible jump. We used. And you'll probably think of it when I say it too. I mean, we basically used indicator variables.
Oh, slab and spike kind of stuff.
Exactly. That's it.
Yeah.
I couldn't remember the term. That's exactly what we did. We did that primarily, and that's what sort of the eventual model that I showed there. That was Leo's work, that he was basically using slab and spike priors to come up with that final model. The other thing that we did that I didn't show here because I knew I was going to run late as it was, and I thought this was an interesting way to look at models, and I hadn't seen this before, was to look at the process variance estimates for different models. At one point we fit, quite a few, like 600 models or something. And for each of them, we had estimates of process variance for the 4 processes, recruitment and 3 survival. And then so on the x-axis, we had like our, say, 400 models. And on the y-axis, let's say we had the process variance for recruitment, and just plot it and to see how process variance would change between models. So you would think, sort of intuitively, if you've got a good model, the process variance ought to be sort of, maybe you hope, as small as possible. You're explaining the variation in the latent variable, the underlying state, pretty well.
Yeah.
And I thought that was a very useful diagnostic. I couldn't find my plot, or else I might have at least inserted it in this talk.
We sometimes look at, in regression analysis, right? We look at percentage deviance explained or that kind of thing. So it's a sort of similar diagnostic of how actually good is this at really explaining the variation you see. Except
exactly.
this variation is in a latent variable, so it's a little tricky. You could only do that when you had known observation errors. Otherwise, the whole thing would totally confound it.
Exactly. Yeah, you're right. I hadn't really quite thought about that way, but you're right. I mean, like R squared is, 1 minus sum to square error, basically, and standardized. And we're kind of, when you look at just a process variance, that's very similar to do like an R squared. Mm-hmm.
Cool. Thanks.
Yeah, that's something else I'd like to explore a bit more, because it seems like it's a pretty obvious diagnostic. But you're right, you have to have known observation variances for this to even make sense.
Okay, anyone else? No.
Okay.
Okay. Well, thank you very much, Ken. That was really interesting. And I'm going to stop recording now.